Okay, so yes, as Trygve said, I'm a postdoc and I'm working on the Digital project, which aims to help aquaculture move from a reactive to a preemptive research strategy in aquaculture. So today I'll be answering the following questions. So first of all, what is metabolic modeling, which is the type of modeling that we're mainly doing? How can metabolic models help us understand salmon? Who are we in the Digital Cell team and what have we achieved during this project? How does the future look for Digital? And finally, a little bit about what we've learned from being part of Digital Life Norway and what others can potentially learn from us. So I'll start by briefly introducing metabolic modeling to give you an idea of what we're working on. So a metabolic model represents the metabolic network, which consists of metabolites, which are the circles in this diagram, and reactions, which are the arrows. And the function of the network is to convert nutrients that are taken up from the environment, in blue here, into the building blocks and energy that is needed to produce biomass and to grow. And this is represented by a biomass reaction highlighted in green. And there are also byproducts produced that are secreted back into the environment. In salmon, the metabolic network is what transforms the nutrients from the feed into the fillet that we eat. And in the Digital project, we're using omics data to reconstruct and to model this network. We're using our models to learn, first of all, more about salmon metabolism, and also to address key challenges facing the salmon aquaculture industry. And in particular, we want to use our models to find new plant-based feeds based on renewable and readily available resources that provide the salmon with the nutrients that it needs, while also producing a healthy fillet for us humans to eat. Our models also constitute a framework for connecting different types of data from our Digital collaborators in academia and industry. And our goal is to allow aquaculture to move from the current situation, where action is taken as a reaction to something that has already happened, to being able to anticipate what will happen in the future and to take preemptive action. So as you just saw, Digital is a collaboration between many different universities and industry partners. The head of the project is Jon Olav, who is also leading our modeling team at NMBU. We are currently 5 people at NMBU working on modeling. So the head of the team is Jon Olav. Philip is a PhD student, and another PhD student will join shortly. I'm a postdoc. Sahar is our data and model manager, and Håvard is a master student. Currently, we are finalizing the first salmon metabolic model, and we're hoping to submit a paper on this within the next few months. We've built a model of the core metabolism of salmon by mapping enzyme encoding genes from the annotated genome to the reactions that are then catalyzed by the enzymes. And we did this manually, which is very time consuming, but it ensures high quality, which is difficult to achieve with automated methods. And this is also the reason why we decided to first focus on energy and amino acid metabolism, as these are the most important processes for transforming feed into fillet. We also built a salmon-specific biomass reaction, and this is based on the genome, the proteome, and the measured whole body composition of salmon. And this biomass reaction allows us to use our model to predict growth, for example, to simulate the effects of different feeds. And this was work done by me, as well as Maxime, Maria, and Philip. After building the model, we of course needed to validate it to get confidence that it gave a reasonable representation of salmon metabolism, and we did this in several different ways. So first, we clustered our salmon model with other published models of animals based on the reaction contents of the models. And we found that the salmon model was closer to the only published fish model of saberfish than to models of human and other mammals. Second, we used MeMote, which is the community standard for testing models, and we got a high score, which shows that the model is functional as well as well annotated. And, oops, my bad. And yeah, finally, we tested the ability of the model to actually perform a large collection of metabolic tasks. And we found that it performed all tasks within the current scope of the model, so all tasks in amino acid, nucleotide, and energy metabolism. And notably, all amino acids that are known to be essential for salmon are also required to achieve growth in the model. As an example on how we can use this salmon model to make useful predictions for aquaculture, Philip used it to predict limiting amino acids in 2 commercial salmon feeds, one marine feed with fish meal and one plant-based feed with soybean meal. Now the pie charts to the left here show the amino acid composition of the 2 feeds. The first pie charts to the right show the portion of amino acids that can actually be utilized by the salmon according to our model. Now there's always one amino acid that will be limiting for growth, and moving further right in this figure, we remove one such limiting amino acid at a time, so we remove one limitation. And this increases the portion of the feed that can then be utilized by the salmon. Now, what Philip found was that the 3 most limiting amino acids, first of all, differed between the 2 feeds, and our model predicts that supplementation of these amino acids could potentially increase feed utilization by about 5% for the marine feed and about 12% for the plant-based feed. In addition to salmon, we're also increasingly interested in modeling the salmon gut microbiota, which is also very important for feed utilization. In particular, we're interested in understanding why the salmon gut becomes very dominated by a new strain of mycoplasma when the fish moves from freshwater to saltwater. And to address this, we've built not one, but actually an ensemble of 13 different models of this mycoplasma from a metagenome assembled genome, or MAG. These models, again, cluster with published models of other mycoplasma. And using this ensemble approach, using multiple models of the same organism, allows us to account for the effect of the significant uncertainty that we have in our metabolic network structures. And this allows us to improve predictions by taking this uncertainty into account. So to help culture this mycoplasma, which is currently not possible in the lab, we use our models to randomly sample minimal growth media. And this allowed us to rank metabolites by their predicted importance for growth. And we hope that this could help prepare a medium that would allow cultivation. Moving forward, we will focus on expanding our current salmon model to the full genome scale metabolic network, mainly to capture processes such as lipid and glycan metabolism. And we've also started to build models that are specific for different tissues, such as liver, using omics data that has been generated in Digicel. And as I said, we are increasingly interested in the salmon gut, and we are reconstructing more salmon gut microbes, so more salmon gut micrometabolic models from metagenome assembled genomes. And the plan is to use these models in a new project that we just got funding for, in which we will build a spatial-temporal model of the salmon gut. So a model that allows us to simulate dynamics in time and space in collaboration with the lab of Will Harcombe at the University of Minnesota. And finally, we're expanding beyond metabolism as well to model other aspects of salmon physiology. So for example, we have started a collaboration on glycosylation modeling with groups in the US and in Sweden. So this is the last slide. I'll just end by summarizing how we think that we are benefiting from being part of Digital Life Norway. First, and perhaps most importantly, it provides us with connections to the systems biology community and to the industry. Second, it incentivizes collaboration, not only within the project, which is highly collaborative, but also between different projects. For example, we had a workshop with the DCOD project that you will hear about soon. Third, we think the research school, conferences, workshops, etc., are very valuable, especially for developing scientists such as myself and our PhD students. And I haven't been part of this project for very long myself, but speaking to the rest of the group, they said that one of the things that they enjoyed the most was meeting scientists from highly diverse backgrounds in informal settings. And finally, it helps us see things from this perspective of other disciplines and industry, and to ask questions such as, how can we make modeling useful, how to collaborate and balance conflicting interests, and how to balance long-term and short-term goals. And I'll end by highlighting what we think is one of our key strengths, and one thing that we think others can learn from us, which is data code and model management. We have a dedicated data manager in our team, and we use the Fairdown Hub web portal to organize our projects. So along with version control of models and software, this really makes it a lot easier to document the work and to eventually allow others to reproduce our results. Thank you.